The hypothalamus and pituitary gland form a complex interface between the nervous system and endocrine system. The brain can influence the activity of neurosecretory cells, and hormones can influence release of other hormones. Your goals for learning are to study hypothalamic pituitary axis anatomy, to review the hypophyseal portal system, to identify the sources of endocrine and neural control of the hypothalamic pituitary axis, to review thyroid hormone. Here's what you need to know: the difference between a neurotransmitter and a hormone, the difference between somatic and autonomic nervous systems, the role of the hypothalamus in control of the autonomic nervous system. To see definitions of terms, click the bold red words. Recall that the pituitary gland, also called the hypophysis, lies beneath the hypothalamus. It is attached to the hypothalamus by a stalk called the infundibulum. The pituitary gland in humans consists of two lobes, an anterior lobe that is made up of endocrine tissue, and a posterior lobe that is made up of neural tissue. The anterior lobe and a small portion of the infundibulum make up the adenohypophysis, and posterior lobe and most of the infundibulum make up the neurohypophysis. We have reviewed 6 hormones secreted by the anterior lobe of the pituitary. They are thyroid-stimulating hormone, or thyrotropin, follicle-stimulating hormone and luteinizing hormone, the gonadotropins, adrenocorticotropic hormone, or corticotropin, growth hormone, and prolactin. These are considered the classic anterior pituitary hormones because their structures and physiological functions in humans are well understood. 4 of these hormones, TSH, FSH, LH, and ACTH, are called tropic hormones because they cause the release of hormones from other endocrine glands. Drag each tropic hormone over its target tissue. In addition to functions of each hormone that we have already reviewed, the anterior pituitary hormones exert important growth effects on target tissues, keeping them healthy and functional. FSH and LH are produced by the same type of cells. Each of the other anterior pituitary hormones is secreted by a different type of cell. This follows the pattern we've observed for other endocrine glands and tissues that produce more than one hormone. Click Continue to proceed. Understanding the pituitary gland requires careful study of the vascular supply. We have noted that the 6 classic anterior pituitary hormones are released directly into the systemic circulation. The ventral hypothalamic hormones regulate the function of the anterior pituitary cells. They are produced in neurons and travel down the axons to terminals where they are stored. The axon terminals do not extend into the pituitary gland. They end at the base of the hypothalamus. Stimulate the ventral hypothalamic neurons by clicking them. Stimuli that induce action potentials in the ventral hypothalamic neurons cause release of hormones into a capillary bed that drains into a second capillary bed before entering the systemic circulation. These 2 capillary beds are connected by the hypophyseal portal veins, special vessels that connect capillary beds. This design allows small amounts of ventral hypothalamic hormones to be present in high concentrations at the anterior pituitary cells. Recall that most of the ventral hypothalamic hormones cause the secretion of anterior pituitary hormones. The ventral hypothalamic hormones that influence the release of tropic hormones and growth hormone are the first hormones in a series that ultimately regulate the secretion of hormones from target glands. The target glands include the thyroid gland, gonads, adrenal cortex, and liver. It secretes insulin-like growth factor. A ventral hypothalamic hormone controls secretion of an anterior pituitary hormone that stimulates secretion of a hormone from a target gland. The final hormone in the chain acts on target tissues of the body. This kind of complex control system provides many input sites where other hormones and neurons can act to achieve fine control over the endocrine system. Let's look first at the influence of hormones. For each hormone series, negative feedback loops control circulating levels of the target gland hormone. Negative feedback exerted by the target hormone can be directed at the anterior pituitary, the ventral hypothalamus, or both. Target hormones from one series can also influence the secretion of hypothalamic or anterior pituitary hormones from another hormone series. We have looked at negative feedback control for thyroid hormone and cortisol in Topic 2. Let's review them briefly here. Click the pituitary gland to continue. Thyrotropin-releasing hormone acts on cells of the anterior pituitary to increase secretion of thyrotropin, TSH, which in turn acts on the thyroid gland to increase secretion of T3 and T4. The thyroid hormones feed back almost exclusively to the anterior pituitary to inhibit secretion of thyrotropin. Click the T3 and T4 to continue. Thyroid hormone is also essential for anterior pituitary cells to secrete growth hormone. Click GH to continue. Corticotropin-releasing hormone acts on cells of the anterior pituitary to increase secretion of corticotropin, ACTH. Corticotropin acts on the adrenal cortex to increase secretion of cortisol. Cortisol feeds back to both the anterior pituitary to inhibit secretion of corticotropin and to the ventral hypothalamus to inhibit secretion of corticotropin-releasing hormone. The cortisol system is subject to far more dynamic changes than the thyroid system, and all hormones of the cortisol series exhibit a circadian rhythm. Click the pituitary gland to continue. Let's look now at the release of prolactin. Prolactin differs from other anterior pituitary hormones in these important ways. Its primary ventral hypothalamic drive is inhibitory. It does not stimulate release of another hormone from a target gland. Prolactin is secreted in small amounts in both males and females. The amount in adult females is slightly higher than the amounts in males and children because estrogen modulates its secretion. Estrogen increases prolactin secretion, but blocks the effects of prolactin on milk production. For example, the high levels of estrogen during pregnancy cause large increases in circulating prolactin. Prolactin, estrogen, and other hormones promote growth of the breasts, but the high levels of estrogen inhibit milk production. Milk production begins after childbirth when estrogen levels drop due to the removal of the placenta. Click the pituitary gland to see a summary diagram. In summary, endocrine control of the hypothalamic pituitary axis can be exerted by negative feedback from a target hormone to a hormone of its own series, modulation by a target hormone of one series to hypothalamic pituitary hormones of another series. The hypothalamus is the highest level integrating center for the autonomic nervous system and the coordinating center for neuroendocrine interactions. Fibers from many parts of the brain influence the endocrine system via hypothalamic neurons. The cerebral cortex is widely connected, both directly and indirectly, to the hypothalamus. Parts of the limbic system, thalamus, basal nuclei, reticular formation of the brainstem, and the retina project to the hypothalamus. Strong emotions like fear, body housekeeping signals like ingestion of food, harmful stimuli that produce pain, trauma, or infection, environmental changes like extreme cold, and light signals from the retina can influence the endocrine system through these circuits. Click the hypothalamus to see a neuroendocrine interaction. Neuroendocrine reflexes like the milk ejection reflex or milk letdown are mediated in the hypothalamus. Suckling induces release of oxytocin that causes the myoepithelial cells of the mammary glands to contract and release milk. The sound of a baby crying can cause a similar letdown of milk, and anxiety and worry can inhibit the milk ejection reflex. Click the hypothalamus again to observe responses of chemically sensitive neurons. Hypothalamic neurons, including the neurosecretory cells that produce vasopressin, behave like osmoreceptors. They detect osmotic changes in body fluids. For example, an increase in the osmolarity of body fluids due to fluid loss, excessive sweating, or diarrhea excites osmoreceptors and stimulates both synthesis and release of vasopressin. Vasopressin acts on the kidneys to promote water reabsorption in an effort to restore normal osmolarity. Click the hypothalamus again to observe circadian rhythms. Neurons of the hypothalamus are pacemakers that generate circadian patterns of hormonal release by the hypothalamic pituitary axis. Circadian rhythms are approximately 24 hours long. The timing of the rhythm is synchronized to the 24-hour Earth day by external signals. We have seen in Topic 2 that cortisol and the hypothalamic pituitary hormones in its series exhibit a circadian rhythm that is synchronized by light. To review what you have already learned about thyroid hormone and to expand your understanding of it, click an image. Thyroid hormones enter cells by diffusion. Recall that 20 times more T4 than T3 is secreted from follicle cells. By converting T4 to T3, the liver and kidney produce about 80% of the free circulating T3. Other target tissues also produce T3, the active form of the hormone. Nearly every cell in the body requires thyroid hormone for normal function. T3 binds to nuclear receptors that alter gene expression. Remember that the thyroid hormones are lipid-soluble and therefore bind to carrier proteins for transport through the circulatory system. Approximately 99% of thyroid hormones are bound, and they exist in dynamic equilibrium with 1% of unbound or free hormone in the plasma. Only the free hormones are available to leave the bloodstream and act on target cells. There is a large circulating reservoir of thyroid hormones. Thyroid hormones have a long half-life in part because they are bound to carrier proteins. Thyrotropin, or thyroid-stimulating hormone, controls secretion of thyroid hormone and promotes growth of the thyroid gland. Thyrotropin-releasing hormone is essential for secretion of thyroid-stimulating hormone. Thyroid-releasing hormone is secreted in a circadian rhythm. Stress may inhibit its release, but there are no known factors in adults that control its secretion. A particular level of thyroid hormone seems to be required for many normal physiological processes. Thyroid hormone modulates physiological processes but does not initiate them. Relatively constant levels of T3 and T4 are maintained by a negative feedback loop that inhibits secretion of thyroid-stimulating hormone. Recall that thyroid hormone includes triiodothyronine, T3, and thyroxine, T4. Each is synthesized from tyrosine and stored in the thyroid follicles. Dietary iodine is essential for normal production of thyroid hormone. T3 has 3 iodine atoms, and T4 has 4 iodine atoms. Thyroid hormone is the most important factor controlling basal metabolic rate. It increases BMR, increasing oxygen consumption and heat production. Thyroid hormone is the main regulator of metabolism. It increases carbohydrate use, is necessary for lipogenesis, amplifies fat mobilization, and increases both synthesis and breakdown of protein. Thyroid hormone is essential for growth. It is permissive for the secretion of growth hormone and for the actions of insulin-like growth factor on bone and other tissues. Thyroid hormone is essential for normal development of the nervous system. Absence of thyroid hormones during the fetal period and immediate postnatal months produce a form of mental retardation called cretinism. If babies who lack thyroid hormone are treated immediately postnatally, the condition can be prevented. If babies are untreated, the condition is permanent and irreversible. Thyroid hormone is essential for normal function of the nervous system in adults. Absence of or low levels of thyroid hormone lead to slowed mental processes. Thyroid hormone amplifies the activity of the sympathetic nervous system by upregulating beta-adrenergic receptors for epinephrine and norepinephrine. Thyroid hormones are broken down by stepwise removal of iodine atoms in peripheral tissues, especially liver and kidney. One way that scientists learn about the endocrine system is to observe the symptoms of patients who secrete too much or too little of a particular hormone. Diseases associated with the thyroid gland are common. On this page, we will study the effects of hypo and hypersecretion of thyroid hormone. Recall the metabolic functions associated with thyroid hormone and use that knowledge to sort the following symptoms. If the symptom is associated with too little TH, drag it to the left side of the page. If the symptom is associated with too much TH, drag it to the right side of the page. Recall the actions of TH on the adult nervous system and use that knowledge to sort the following symptoms. Full-blown hypothyroid syndrome in adults is called myxedema. The term comes from the edematous, puffy appearance of patients due to accumulation of water-holding carbohydrates in the skin. Hyperthyroidism is most commonly caused by an autoimmune disease called Graves' disease. The body produces an antibody that mimics the action of thyroid-stimulating hormone. The thyroid gland responds by enlarging and secreting increasing amounts of thyroid hormones. A unique symptom of Graves' disease is protrusion of the eyeballs, a condition called exophthalmos, which results from deposits of water-holding carbohydrates behind the eyes. Click the Graves' disease patient to continue. To better understand the thyroid hormone pathologies, we need to know whether the hypo or hypersecretion of thyroid hormone is a primary effect due to changes in the gland itself, or whether the effect is secondary to changes in thyroid-stimulating hormone. Click Continue to proceed. Let's look at primary hypothyroidism first. It results from failure of the thyroid gland to secrete thyroid hormone or failure of the thyroid gland to secrete enough thyroid hormone due to a deficiency of dietary iodine. In both cases, the amount of T3 and T4 secreted diminishes. Recall the negative feedback inhibition exerted on the anterior pituitary by thyroid hormone. As plasma levels of thyroid hormone fall, the inhibition to the anterior pituitary is interrupted, and the anterior pituitary secretes increasing amounts of TSH. Remember that TSH stimulates growth of the thyroid gland, therefore high levels cause enlargement of the gland. This is called a goiter. When the hypothyroidism is associated with lack of dietary iodine, it is called an iodine-deficient goiter. Such goiters used to be common in parts of the Midwestern United States due to low iodine concentrations in the soil. The introduction of iodized salt helped reduce both the incidence of iodine-deficient primary hypothyroidism and the number of such goiters. Click the image of the patient with the goiter to continue. Secondary hypothyroidism results from failure of the anterior pituitary or hypothalamus. In this condition, when thyroid-stimulating hormone, and possibly also thyrotropin-releasing hormone, levels are low, T3 and T4 levels diminish. Because TSH levels are low, secondary hypothyroidism will not be associated with enlargement of the thyroid gland. Click Continue to see a summary of hypothyroidism. Let's look now at primary hyperthyroidism. It results from a thyroid hormone-secreting tumor of the thyroid gland, or Graves' disease. In both cases, the amount of T3 and T4 secreted increases. Recall the negative feedback inhibition exerted on the anterior pituitary by T3 and T4. As plasma levels of the thyroid hormone rise, the inhibition to the anterior pituitary increases, and the anterior pituitary secretes decreasing amounts of TSH. Because of the hormone pattern observed, we expect that primary hyperthyroidism will not produce a goiter. With low TSH and the concomitant loss of its tropic actions on the thyroid gland, we might expect the thyroid gland to remain the same size or atrophy. That is the case for thyroid hormone-secreting tumors. In Graves' disease, the body produces an antibody called thyroid-stimulating immunoglobulin, TSI, that mimics the actions of TSH. The high levels of thyroid-stimulating immunoglobulin stimulate growth of the thyroid gland and can produce a goiter. Note that increased levels of thyroid hormone do not diminish production of TSI because there is no negative feedback relationship between them. Click the image of the patient with the goiter to continue. Secondary hyperthyroidism results from excess secretion by the anterior pituitary or hypothalamus. In this condition, when thyroid-stimulating hormone, and possibly also thyroid-releasing hormone, levels are high, T3 and T4 levels increase. Because TSH levels are high, secondary hyperthyroidism will be associated with enlargements of the thyroid gland. Click Continue to see a summary of hyperthyroidism. Here's a summary of what we've covered. Hypothalamic neurons produce hormones. Some hypothalamic hormones enter the systemic circulation from the posterior pituitary, and others influence the anterior pituitary via the hypophyseal portal system. TSH, FSH, and LH, ACTH, and GH from the anterior pituitary influence secretion of a third hormone from a target gland. Target gland hormones can exert negative feedback control of the hypothalamus and/or anterior pituitary. Neurons throughout the brain influence the hypothalamus. Hypo and hypersecretion of thyroid hormone cause widespread metabolic consequences throughout the body. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.